Hello everyone. This section is for measurement of microbial population size. After learning this section, you should be able to evaluate direct cell counts, variable counting methods, and cell mass measurements for determining population size, and explain why plate count results are expressed in terms of colony-forming units. There are many ways to measure microbial growth to determine growth rate constants and generation times. Either population number or mass may be followed, because growth leads to increases in both. In this section, let's talk about direct measurement of cell numbers and variable counting methods first. Let's say the direct measurement of cell numbers. We use counting chamber in this method. The counting chamber consists of specially designed slides and cover slips. The space between the slide and cover slip creates a chamber of known depth. You can see it on the right. On the bottom of the chamber is an etched grid that facilitates counting the cells. The number of microorganisms in a sample can be calculated by taking into account the chamber's volume and any dilutions made of the sample before counting. One disadvantage of using counting chambers is that to determine population size accurately, the microbial population must be relatively large and evenly dispersed, because only a small volume of the population is sampled. Then let's say variable counting method. Several plating methods can be used to determine the number of variable microbes in a sample. These are referred to as either variable counting methods or plate counts, because they count only those cells that are able to reproduce when cultured. We have two commonly used procedures. They are spread plate and the pour plate techniques. Firstly, let's see the spread plate technique. Usually, a small amount of the sample is pipetted to the center of a solidified media. Then we use a glass spreader. After the glass spreader is sterilized, by dipping it into ethanol and briefly flaming it. Then the spreader is cooled and then used to spread the sample evenly over the surface of the media. Then we can get colonies on the plate. The second is pour plate technique. Firstly, we dilute the samples for several times, and some of the dilutions are mixed with warm egg and poured onto the plates. Isolated cells grow into colonies on the surface. These colonies appear round, and within the media appear lens-shaped. The isolated colonies can be counted or used to establish pure culture. When using these methods to determine population size, the samples should be yield between 25 and 250 colonies per plate for accurate counting. Once the number of colonies is known, the original number of viable microorganisms in the sample can be calculated from that number and the same dilution. For example, If 1 mm of a solution diluted by a factor of 1 times 10 to the 6 yield 115 colonies. For example, 1.5 times 10 to the second colonies. Then the original sample contains around 1 times 10 to the 8th cells per millimeter. Because it is not possible to be certain that each colony arose from an individual cell, the results are often expressed in terms of colony-forming units, CFU for short, rather than the number of microorganisms. Direct measurement of cell numbers and variable counting methods are common methods for most microorganisms, but for some special samples, they are not suitable. Then let's say the membrane filtration procedure. It is used to measure the bacteria in aquatic samples. Look at the picture. It shows the membrane filtration procedure. Firstly, put a membrane filter on a filter support. Then the water sample filters through the membrane filter, and the membrane filter removed and placed in plate containing the appropriate media. After incubation for 24 hours, then you can get the typical colonies. Plating techniques are simple, sensitive, and widely used for variable counts of bacteria and other microorganisms in samples of food, water, and soil. However, several problems can lead to inaccurate counts. First, the population will be underestimated if clumps of cells are not broken up, and microorganisms will disperse. The hot agar used in the pour plate technique may injure or kill sensitive cells. Thus, spread plates sometimes give higher counts than pour plates. Plate counts will be artificially low if the media employed cannot support growth of all the variable microorganisms present. Then let's talk about the measurement of cell mass. Techniques for measuring changes in cell mass also can be used to determine population size. First, determination of microbial dry weight. Cells growing in liquid media are collected by centrifugation, washed, dried in an oven, and weighed. This is an especially useful technique for measuring the growth of filamentous fungi. However, it is time-consuming and not very sensitive. We often use spectral photometry. When the concentration of bacteria reaches about 1 million cells per millimeter, the media appears slightly cloudy or turbid. Further increases in concentration result in great turbidity, and less light is transmitted through the media. You can see it on the picture. The picture shows turbidity and microbial mass measurement. The extent of light scattering can be measured by a spectral photometer and is called the absorbance or optical density. Absorbance is almost linearly related to cell concentration at absorbance levels less than about 0.5. If the sample exceeds this value, it must first be diluted and then absorbance measured. Thus, population size can be easily measured as long as the population is high enough to give detectable turbidity. Okay, that's all for this section. Here is a word list for you.